A discovery does not usually rewrite the history of humanity. Most archaeological finds add another piece to an already familiar puzzle. But in 2008, deep inside a limestone cave in the Altai Mountains of southern Siberia, archaeologists recovered something so small that it could almost fit on the tip of a finger. It was exactly that. A tiny fragment of the little finger bone from a young individual who had lived tens of thousands of years ago. There was no complete skeleton, no skull, no face to reconstruct, no extraordinary burial. At first glance, it seemed like one more anonymous fossil among thousands that had already been excavated across Eurasia. Yet, within only 2 years, this insignificant fragment would force scientists to redraw the human family tree and admit that an entire branch of humanity had existed without anyone knowing it. The shock was not simply that another ancient human had been discovered. Archaeology had revealed new human species before. The true surprise was that this one was identified almost entirely through its DNA. Before researchers even understood what this individual looked like, genetic evidence revealed that it belonged neither to modern humans nor to Neanderthals. It represented a population completely unknown to science. For the first time in history, genetics had discovered a lost human species before traditional archaeology had done so. That single breakthrough changed far more than the story of one cave. It challenged decades of assumptions about how many kinds of humans once walked across Eurasia. To understand why this discovery was so revolutionary, it is necessary to look at what scientists believed before the Denisovans entered the picture. For much of the 20th century, the late human story seemed relatively straightforward. After more than a century of fossil discoveries across Europe, Asia and Africa, researchers had assembled what appeared to be a coherent timeline. Modern Homo sapiens evolved in Africa roughly 300,000 years ago. Neanderthals developed across Europe and Western Asia after diverging from a common ancestor hundreds of thousands of years earlier. By approximately 60 to 70,000 years ago, modern humans expanded out of Africa into Eurasia, where they encountered Neanderthals. Eventually, Neanderthals disappeared around 40,000 years ago, leaving Homo sapiens as the only surviving human species. Although scientists debated exactly how often these populations interacted, the overall picture remained surprisingly simple. In Eurasia, there were essentially two major human groups during the late Pleistocene, ourselves and the Neanderthals. Fossils discovered in France, Germany, Croatia, Iraq, Israel and elsewhere could almost always be assigned to one of those two lineages. Every new excavation was expected to fit somewhere within that framework. This model became even stronger during the 1990s and early 2000s as ancient DNA technology began transforming paleoanthropology. In 1997, researchers led by Svante Paabo successfully extracted mitochondrial DNA from a Neanderthal fossil found in Germany's Neander Valley. It marked the first successful recovery of ancient human DNA and proved that genetic material could survive for tens of thousands of years under exceptional conditions. Over the following decade, laboratories improved extraction techniques, while contamination controls became increasingly sophisticated. Fossils were no longer studied only through their bones. Scientists could now compare entire genomes. By 2010, the Max Planck Institute for Evolutionary Anthropology in Leipzig had completed the first draft genome of a Neanderthal. The results confirmed that Neanderthals and modern humans had interbred. Today, people outside Africa carry roughly 1 to 2% Neanderthal DNA, providing direct evidence that these populations did not simply replace one another, they exchanged genes before Neanderthals vanished. For many researchers, this seemed like the final missing chapter. The evolutionary relationships between modern humans and Neanderthals were becoming increasingly clear. Ancient DNA was confirming what fossils had suggested for decades. It appeared that the major questions had largely been answered. But there was one problem. The fossil record itself had always been incomplete. Human bones are remarkably rare. Most individuals leave no trace at all. Skeletons decay quickly unless they are buried under highly unusual environmental conditions. Even when fossils survive, they are often broken into scattered fragments by animals, geological activity, or thousands of years of erosion. Entire populations may disappear without leaving behind a single complete skeleton. Paleoanthropologists have long understood that every fossil discovery represents only a tiny sample of ancient reality. Imagine trying to reconstruct the population of an entire continent. using only a handful of broken bones recovered over hundreds of thousands of years. The gaps are enormous. Scientists accepted those limitations because there was little alternative. They could only study what nature had preserved. Still, few believed those missing pieces concealed an entirely unknown human lineage. By the early 21st century, Denisova Cave had already attracted archaeological attention for a different reason. Located in Russia's Altai Mountains near the borders of Kazakhstan, Mongolia, and China, the cave preserved exceptionally stable temperatures throughout the year. Those cold conditions protected organic material unusually well, making it one of the best archaeological sites in northern Asia. Excavations had uncovered stone tools, animal remains, ornaments crafted from bone and ivory, and evidence that different groups of humans had occupied the cave across hundreds of thousands of years. Yet nothing suggested that the site contained evidence of an undiscovered branch of humanity. Then, during the 2008 excavation season, archaeologists uncovered one tiny bone fragment from layer 11. It measured only a few centimeters in length and belonged to the little finger of a juvenile, estimated to have been between 5 and 13 years old. Morphologically, the fragment revealed almost nothing. It could not confidently be identified as Neanderthal. It could not confidently be identified as Homo sapiens. There simply was not enough anatomy preserved. Under previous generations of archaeology, the story might have ended there. The bone would likely have been catalogued, stored in a museum collection, and largely forgotten. Instead, researchers decided to do something that would change human history forever. They extracted its DNA. Extracting DNA from ancient human remains is one of the most demanding procedures in modern science. Every surface, every tool, and every researcher represents a possible source of contamination. Modern human DNA is everywhere. A single skin cell or an unnoticed breath can overwhelm genetic material that has survived inside a fossil for tens of thousands of years. Before any sequencing began, the tiny finger bone from Denisova Cave was cleaned under sterile laboratory conditions at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. Researchers removed its outer surface, drilled into the dense interior, and carefully collected bone powder that still contained fragments of ancient DNA. Years of advances in sequencing technology had finally made it possible to recover genomes from fossils once considered too degraded to study. The team was led by geneticist Svante Paabo, whose laboratory had already transformed paleoanthropology by recovering and sequencing Neanderthal DNA. At first, no one expected another revolution. The simplest assumption was that the finger bone belonged to a Neanderthal because Neanderthal remains had already been found in the Altai region. If that proved correct, the specimen would simply become another valuable addition to an expanding Neanderthal genetic database. Instead, the first genetic results created immediate confusion. The mitochondrial DNA did not match Homo sapiens. That alone was not surprising. It also failed to match Neanderthals. That was almost impossible to explain. Rather than sitting inside the known genetic variation of either species, the sequence occupied an entirely different branch of the human evolutionary tree. At first, researchers considered every possible explanation: laboratory contamination, sequencing errors, damaged DNA, a pathological individual. Any ordinary explanation seemed more likely than the existence of an unknown human population. The analyses were repeated again and again. Each time the conclusion remained the same. In December 2010, the findings were published in the journal Nature. The paper announced that the fossil represented a previously unknown group of archaic humans that had shared a common ancestor with Neanderthals, but had evolved along a separate lineage for hundreds of thousands of years. The scientific community had expected new fossils. It had expected refinements to existing evolutionary models. It had not expected an entirely new branch of humanity to emerge from a fragment of bone barely large enough to fit inside a laboratory vial. The implications reached far beyond one cave in Siberia. For more than a century, paleoanthropologists had relied primarily on anatomy. Every newly discovered fossil was identified through its physical characteristics. Skull shape, jaw structure, teeth, limb proportions, and countless other anatomical details formed the foundation of human evolutionary research. The Denisova finger bone contained almost none of those clues. If researchers had depended solely on morphology, the fossil might never have received special attention. It was genetics that revealed its true identity. For the first time, scientists had discovered a human population through DNA before they understood what its members actually looked like. This marked one of the greatest methodological shifts in the history of archaeology. The finger bone itself belonged to a young girl, later designated Denisova III. Beyond her age, almost nothing could be determined from the fossil alone. There was no skull to reconstruct facial features, no pelvis to estimate body proportions, no complete skeleton to reveal height or locomotion. Ironically, researchers knew more about her evolutionary relationships than about her appearance. They could trace her ancestry across hundreds of thousands of years while remaining unable to say whether her face resembled a Neanderthal, a modern human, or something entirely different. As sequencing technologies improved, the team recovered far more than mitochondrial DNA. They reconstructed a high-quality nuclear genome from the same fossil, making it one of the oldest and most complete ancient genomes ever analysed. Nuclear DNA confirmed that Denisovans shared a common ancestor with Neanderthals approximately 400,000 years ago, after both groups separated from the lineage leading to Homo sapiens. Yet after that split, Denisovans followed their own independent evolutionary path for hundreds of thousands of years. This discovery created an unexpected paradox. If Denisovans had existed as a distinct population for such an immense span of time, why had no one recognized them before? How could an entire branch of humanity remain invisible until 2010? The answer exposed one of archaeology's greatest limitations. Scientists had assumed that the fossil record represented most major human populations. In reality, it represented only the fossils fortunate enough to survive. A single cave with exceptional preservation conditions had revealed a lineage that might otherwise have remained unknown forever. Countless other populations could have vanished without leaving complete skeletons for future researchers to discover. The evolutionary tree that appeared so well understood suddenly looked incomplete again. Even more surprising evidence soon emerged from Denisova Cave itself. Researchers recovered several unusually large molar teeth from the same archaeological layers. Unlike the finger bone, these fossils displayed anatomical features unlike either modern humans or Neanderthals. Their crowns were exceptionally broad, while the roots showed primitive characteristics rarely seen in later human species. Genetic analysis confirmed that these teeth also belonged to Denisovans. Although only a handful of fossils had been identified, each new specimen strengthened the conclusion that this was not an isolated individual, but a long-lived human population. Yet despite these discoveries, Denisovans remained strangely invisible. Scientists now possessed one of the highest quality ancient genomes ever recovered, but they still lacked something that every other major human species possessed: a recognizable face. No complete skull had ever been found. No artist could confidently reconstruct their appearance. Their existence was undeniable, but their identity remained elusive. And then the mystery became even stranger. When researchers compared the Denisovan genome with the DNA of living people around the world, they found something no one had predicted. The descendants of this forgotten population were not entirely gone. Millions of people alive today were still carrying pieces of Denisovan DNA. The discovery of the Denisovan genome solved one mystery, but it immediately created another. If an entirely unknown branch of humanity could remain hidden until the 21st century, how many others had escaped the fossil record altogether? For decades, archaeologists had accepted that fossils represented the foundation of human evolution. Every new skull, every jawbone, every tooth helped fill another gap in humanity's past. But Denisova Cave demonstrated something profoundly unsettling. A human population could survive for hundreds of thousands of years, spread across enormous regions, interbreed with other humans, and still leave behind almost nothing that could be recognized through traditional archaeology. Suddenly, scientists began looking at ancient DNA in a completely different way. Instead of asking what fossils could tell them about genetics, they started asking what genetics could reveal without fossils. The answer was astonishing. As researchers sequenced the genomes of present-day populations from around the world, they discovered patterns that could not be explained by any known human species. Certain stretches of DNA appeared far older than expected. They did not belong to Homo sapiens, they did not match Neanderthals, they did not match Denisovans either. They belonged to no one. Geneticists began referring to these mysterious contributors as ghost populations. The name did not imply supernatural beings, it described ancient human groups that had left genetic descendants, but no identifiable fossils. Their bodies had disappeared from the archaeological record, yet fragments of their genomes continued living inside modern people. In other words, evolution had left fingerprints without leaving a body. One of the strongest examples emerged in Africa. Because ancient DNA degrades rapidly in warm climates, recovering genetic material from African fossils remains extremely difficult. Yet modern African genomes contain signals suggesting that Homo sapiens may have interbred with one or more archaic human populations that scientists have never identified. Several studies published over the last decade, including work by researchers at the University of California, Los Angeles, and the University of Pennsylvania, suggest that these unknown populations diverged from the ancestors of modern humans hundreds of thousands of years ago before later mixing back into our lineage. No skull has ever been confidently assigned to these populations. No complete skeleton has been recovered. They exist only because mathematics hidden inside modern genomes says they must have existed. It is one of the strangest situations in science. Researchers know these humans once lived. They simply do not know who they were. Denisovans proved that such discoveries were possible. Ghost populations suggested they might be common. The human family tree was beginning to resemble less of a simple branching diagram and more of an intricate network, with populations separating, migrating, meeting again, and exchanging genes repeatedly over hundreds of thousands of years. Then another extraordinary discovery pushed the Denisovan mystery even further. When scientists compared Denisovan DNA with the genomes of living people, they expected to find traces among populations living closest to Siberia. Instead, they found almost the opposite. Most Europeans carried virtually no Denisovan ancestry. East Asians possessed only a very small amount. The highest percentages appeared thousands of kilometers away among the indigenous peoples of Papua New Guinea, Aboriginal Australians, and many Melanesian island populations. In some individuals, Denisovan DNA accounts for between 3 and 6% of the genome, far exceeding the amount found anywhere else on Earth. This result made almost no geographical sense. The only confirmed Denisovan fossils had been discovered inside a cave in southern Siberia. How could their strongest genetic legacy survive on tropical islands near Australia? There were only two possible explanations. Either Denisovans had once occupied an enormous range stretching across much of Asia, or there had been multiple Denisovan populations living in different regions over vast periods of time. Evidence increasingly supports the second possibility. Genetic analyses indicate that Denisovans were probably far more genetically diverse than Neanderthals. Some scientists now believe the name Denisovan may actually describe several closely related populations rather than one uniform species. Different Denisovan groups may have adapted to forests, mountains, tropical environments, and northern climates, occupying regions from Siberia to Southeast Asia long before modern humans arrived. Remarkably, modern archaeology has begun uncovering clues that support this idea. A fossilized jawbone discovered in Baishia Karst cave on the Tibetan plateau was originally impossible to classify because no usable DNA survived. However, in 2019, researchers analyzed ancient proteins preserved inside the jaw using paleoproteomics. The results, published in Nature, identified the fossil as Denisovan. Dating suggests it is at least 160,000 years old. This discovery transformed scientists' understanding of Denisovan geography. They were no longer confined to one cave. They had already adapted to one of the harshest environments on Earth long before Homo sapiens reached the Tibetan plateau. Yet perhaps the most remarkable inheritance they left behind was not another fossil. It was a biological advantage still carried by millions of living people. Among modern Tibetans, scientists identified a variant of the EPAS1 gene that allows the body to function efficiently in low-oxygen environments at elevations exceeding 4,000 meters. Instead of producing excessive red blood cells, which can become dangerous at high altitude, carriers regulate oxygen use far more effectively. In 2014, studies published in Nature showed that this version of EPAS1 did not originate in Homo sapiens. It came from Denisovans. Thousands of generations after the Denisovans themselves disappeared, one of their genetic adaptations continues helping people survive in conditions where many others struggle simply to breathe. The species may be gone, but parts of their biology are still alive. And just when scientists thought the Denisovan story could not become any stranger, another tiny bone fragment from Denisova Cave revealed evidence of something that had never before been observed directly in the history of human evolution. It belonged to the daughter of two different human species. In 2018, another tiny fragment of bone recovered from Denisova Cave delivered one of the most extraordinary discoveries in the history of paleoanthropology. Like the famous finger bone, this specimen revealed almost nothing through its physical appearance. It was a small piece of long bone belonging to a young female who had died at around 13 years of age, more than 90,000 years ago. On its own, the fossil appeared completely ordinary. Its DNA told a very different story. Researchers at the Max Planck Institute sequenced the genome and found something they had never expected to see. Half of her DNA came from a Neanderthal. The other half came from a Denisovan. This individual, known informally as Denny, was the direct offspring of two different human groups, not a distant descendant. Not evidence of ancient mixing inferred through statistical models. Her mother was a Neanderthal. Her father was a Denisovan. This represented the first confirmed first-generation hybrid ever discovered among ancient humans. The finding was published in Nature in 2018, immediately becoming one of the strongest pieces of evidence that different human populations did not simply replace one another when they met. They formed families. For decades, researchers had known that modern humans carried small amounts of Neanderthal DNA. They had also identified Denisovan DNA in populations across Asia and Oceania. Those discoveries demonstrated that interbreeding had occurred sometime in the distant past. Denny proved something even more remarkable. These encounters were not isolated accidents. If scientists happened to discover a first-generation hybrid from only a handful of Denisovan fossils, then such unions were probably not exceptionally rare. Statistically, the probability of finding a direct hybrid among so few individuals suggests that Neanderthals and Denisovans likely encountered each other repeatedly over thousands of years. Even more surprising was the genetic history of Denny's father. Although he was genetically classified as a Denisovan, researchers found evidence that one of his own ancestors had inherited Neanderthal DNA several hundred generations earlier. The boundaries between these populations were far more fluid than anyone had imagined. Instead of separate branches living in complete isolation, Eurasia appears to have been inhabited by overlapping populations that occasionally met, exchanged genes, separated again, and repeated the process across tens of thousands of years. The traditional image of human evolution as a simple sequence of replacement began to collapse. It was becoming a network rather than a straight line. Yet despite all these breakthroughs, Denisovans remain among the least understood members of our evolutionary family. Scientists possess one of the highest quality ancient genomes ever recovered. They have identified multiple Denisovan individuals. They have confirmed their relationship to Neanderthals. They know Denisovans contributed genes to millions of living people. They even know that some of those inherited genes continue helping humans survive in extreme environments today. And still, they cannot answer some of the most basic questions. What did Denisovans actually look like? No complete skull has ever been recovered. Without one, every facial reconstruction remains largely speculative. In 2024, researchers used artificial intelligence and genomic information to predict possible Denisovan anatomical features, suggesting a broad face, large teeth, and a wide rib cage. While these reconstructions generated considerable interest, they remain computational models rather than direct evidence. Until more complete fossils are discovered, their true appearance cannot be confirmed. Scientists also do not know how many Denisovan populations existed. Genetic analyses increasingly suggest that the Denisovans found in Siberia were only one branch of a much larger and more diverse lineage. Different populations may have occupied East Asia, Southeast Asia, and the Tibetan Plateau for hundreds of thousands of years, adapting to dramatically different climates and environments. Some researchers even argue that fossils discovered decades ago but difficult to classify, including remains from China, could eventually prove to belong to Denisovan populations. If confirmed, it would dramatically expand both their geographic range and their evolutionary history. Perhaps the greatest unanswered question concerns their disappearance. Unlike the extinction of the dinosaurs, there was no single catastrophic event. Unlike Neanderthals, whose decline has been studied for decades, Denisovans left so few fossils that scientists cannot reconstruct their final centuries with confidence. Did climate change gradually reduce their populations? Were they absorbed into expanding groups of Homo sapiens through repeated interbreeding? Did competition for resources contribute to their decline? Or did different Denisovan populations disappear at different times for entirely different reasons? At present, no one knows. Every new excavation has the potential to rewrite the story once again. In recent years, researchers have begun recovering ancient environmental DNA directly from cave sediments, even when no visible fossils are present. Genetic traces preserved in soil have already revealed that both Neanderthals and Denisovans occupied Denisova Cave during different periods, demonstrating that ancient DNA can survive even after bones have disappeared. This technology is opening an entirely new chapter in archaeology, allowing scientists to detect vanished human populations without finding a single skeleton. It raises an extraordinary possibility. How many other caves around the world contain invisible genetic evidence of humans that have never been identified? How many forgotten populations are still hidden beneath layers of sediment, waiting for technology rather than excavation to reveal their existence? Only a generation ago, scientists believed they understood the final chapters of human evolution. Eurasia belonged to Neanderthals and Homo sapiens. The story appeared complete. Then a fragment of a child's finger changed everything. Today, Denisovans are no longer a hypothesis. They are a confirmed branch of humanity. They shared a common ancestor with Neanderthals, adapted to environments ranging from Siberia to the Tibetan Plateau, exchanged genes with our own ancestors, and left a biological legacy that survives in millions of people across the world. Yet they also remind us how incomplete our understanding still is. Nearly every fact we know about them comes from a handful of fossils recovered inside a single cave and a few additional discoveries scattered across Asia. That is an astonishingly small amount of evidence for a population that survived for hundreds of thousands of years. Perhaps the greatest lesson of the Denisovan discovery is not that scientists found another human species, it is that they found one they never expected to exist. If an entire branch of humanity remained invisible until 2010 despite more than a century of archaeological research, then the history of our species is almost certainly not finished. Somewhere beneath layers of rock, frozen ground, or ancient cave sediments, there may still be other forgotten chapters of the human story waiting to be uncovered. The next discovery may not simply answer another question. It may force us to rewrite the history of humanity all over again.